Hi everyone, this is Shakil Ahmed, your biology educator, and here we are with the second part of our previous video where we discussed paper 1 variant 13 of winter 2023. We discussed question numbers 1 to 20 in that part of the video, and this is the second part where we'll discuss the last 20 questions from 21 to 40 onwards. So question number 21, the electron micrograph shows a cell in the stage of the mitotic cycle. Which statement explains why it is difficult to identify the stage of the mitotic cycle shown? Chromosomes have supercoiled and are visible, but centrioles are not visible. We cannot see chromosomes anywhere in this photomicrograph. That means this option is wrong. Anaphase may be continuing or telophase may be starting. If we go back to the concept of the anaphase, we know that in the case of anaphase, the chromosomes move to the opposite poles, and we can clearly see here that the chromosomes have moved to the opposite poles. So this can be the end of anaphase. And in the telophase, when the chromosomes have reached the opposite poles, we see the reformation of the nuclear membrane, the nucleus takes place. So this can be the start of the telophase as well. So this option may be correct. It's unclear whether the electron micrograph shows two cells in the metaphase. We cannot see two cells here, so this is also wrong. Some people may consider interphase to have started. In case of interphase, the cell has a nucleus, and nucleus is not visible in this cell. So option B is the correct one. Move to question number 22. During lagging strand replication, short fragments of DNA are produced. The fragments are joined together by DNA linkage. Which row correctly describe the structure of these short fragments of DNA? We know DNA molecule contains the two strands. One is the leading strand which contains the gene, and the other is the lagging strand. So when both these strands are copied, we will see that the new polynucleotide chains will be formed. One with this, one with this. I am talking about the new polynucleotide chains. So let's go to the question. Have base sequences that are complement to the newly synthesized leading DNA strand? They are talking about the lagging strand. Obviously, the base sequences here will be complementary to the base sequences here. So this is the correct option. Consists of one polynucleotide strand. The new strand only is one polynucleotide. That's also correct. Contains only carbon, hydrogen, oxygen, and nitrogen, which is wrong because DNA is made up of the nucleotides, and nucleotides contain not only carbon, hydrogen, oxygen, and nitrogen, they also contain phosphate with them. So this is write it only, that means this is the wrong option. So first two are correct, last one is wrong. Option D is the correct one. The genome of the bacterium Escherichia coli has been altered to enable it to code for an amino acid that's not found in nature. All the ATC, DNA stop triplets on the strand of DNA that's transcribed, have been substituted to ATT. So that means there is no stop triplets have been altered. The ATC triplet can then be inserted. So they insert the ATC triplet to code for the new amino acid. A new transfer RNA can then be considered to carry the new amino acid. What is the anticodon of this new transfer RNA? We know the anticodon on a transfer RNA has the same sequence as that of the DNA, except that on the RNA we will have U, on DNA we will have T. So if it is ATC, that is the anticodon will be ATC, it will be AUC. So B is the correct option. Question number 24. 40% of the bases in a section of a non-transcribed strand of DNA are purine bases. Purine molecules. What will be the total percentage of cytosine and uracil bases in the primary transcript that's transcribed from the other strand of the DNA? We know the cytosine and uracil, both of them are the pyrimidines, because adenine and guanine are the only purines. And if purines are 40%, that means the pyrimidines will be 60%. So D is the correct answer. Which statement about xylem vessel elements is correct? Hollow vessels enable the constant movement of water up and down upward. The xylem vessels are not hollow. Pits enable the movement of water into the adjacent xylem vessels. This seems correct. Why? Because we know the pits are basically the plasmodesmata between the cells, and there is no lignin deposition at the plasmodesmata. So they allow the movement of water because lignin prevents the movement of water. Vessels contain numerous mitochondria to generate ATP for active transport. We don't have any organelles in the xylem cells, so this is wrong. Vessels contain perforated cross. Perforated cross walls are present in the phloem sieve plates. So that is option B is the correct answer. Ammonium ions NH4 can enter the xylem of plant roots by two pathways. In the apoplast pathway, ammonium ions move through cell walls until this path is blocked by the Casparian strip. Ammonium ions then enter the cytoplasm of root cells. In the symplast pathway, ammonium ions move through the cytoplasm of root cells. A scientist measured the uptake of ammonium ions into the xylem of Arabidopsis thaliana. The SGN3 mutant of Arabidopsis thaliana does not have a Casparian strip. In the SGN3 mutant, ammonium ions can enter the xylem without entering the cytoplasm of the root cells. So the bar charts show the scientist's result. This is uptake of the ammonium ions into xylem. This is at the low ammonium concentration. This is at the high ammonium concentration. This is non-mutant and this is mutant. This is non-mutant, this is mutant. Which conclusion is correct? Fewer ammonium ions enter the xylem and they have to move through the cytoplasm of the root cells. We can see there is not much difference whether it is present, the Casparian strip is present or not. So this is the wrong option. More ammonium ions enter the xylem at the low soil concentration of ammonium ions. Nothing of that sort can be seen from the graph. The Casparian strip acts as a barrier to reduce the movement of ammonium ions in the xylem. We cannot see the removal of the Casparian strip has got any phenomenal effect on the movement of the ammonium ions. The loss of Casparian strip has little effect on the movement of the ammonium ions into the xylem vessel. This is perfectly correct, because if you look at this thing, that there is not much difference between the two graphs, two bars. Question number 27. Which row correctly shows processes required for the movement of water from a root hair cell to the atmosphere? So if we have a root hair cell which absorbs the water, so the water moves from the higher concentration to the lower concentration by osmosis, from higher water portion to the lower water portion, that it moves the xylem through the xylem vessels, and we know that it is the force of cohesion that keeps them together. There is evaporation of water from the surface of the leaves that actually creates the transpiration pull, pulling the water column up. So there is cohesion, this is correct. There is diffusion, there is evaporation. So option A is the correct option. Which process can be carried out by a mature red blood cell? A mature red blood cell can carry out active transport. There is nothing wrong in that. Cell division, no. Mature red blood cell will not undergo any cell division. Phagocytosis is the function of the white blood cells, and protein synthesis will. A red blood cell does not contain nucleus, so there will be no protein synthesis in it. So active transport is the correct option. The photomicrograph shows 3 white blood cells labeled X, Y, and Z. Which row correctly identifies these cells? I can clearly see that Y is a neutrophil because we can see the bilobed nucleus. Then X is a lymphocyte because lymphocytes are smaller than the macrophages or monocytes. So X is a lymphocyte, Y is a neutrophil, and P is a macrophage or a monocyte. So this is the correct option. Z is the monocyte. When a blood vessel is damaged, it can result in the problems in the body. Which row identifies the blood vessel that could have been damaged? Blood is not being distributed to the organs that need it. So that means if blood is supplied to the organs, there is a problem with the muscular artery. Blood is not returning to the heart and is collecting the organs. Blood is collected by the veins, so there is problem with the veins. Blood surges mean that relatively constant blood pressure is not maintained. Maintaining a pressure inside the blood vessels is a work of the elastic fiber or elastic artery. So that means option D is the correct one. Question number 31. The diagram shows how tissue fluid is formed. So this is the dark one, this line is the blood pressure, and the dotted lines is the water potential. So this is the arterial end, this is the venous end. What is the net pressure causing the tissue fluid to flow out of the capillary at the arterial end? So we are talking about the arterial end here. Let's see what the total pressure inside. 4.3 plus and minus 3.3 means it is plus 1.0. What is the pressure outside? So it is 1.1 plus minus 1.3. That means it is minus 0.2. So the pressure difference is 1.0 minus 0.2, which means 1 plus 0.2. That means 1.2 plus is the pressure difference. So 1.2 kilopascal. C is the correct option. What happens when carbon dioxide is transported by red blood cells? Hydrogen ions move into the red blood cells. No, hydrogen and ions move out of the red blood cells. So this is the wrong option. Chloride ions move into the red blood cells and bind to hemoglobin. They don't bind to hemoglobin. Chloride ions move into the red blood cell and hydrogen ions move into the plasma. Chloride ions move into the red blood cell and hydrogen ions are picked up by the hemoglobin. Chloride ions move into the red blood cell and hydrogen and carbonate ions move into the plasma. This is what we call the chloride shift. This is the correct option. What is typically found in trachea, bronchi, bronchioles in the human gas exchange system? Okay, we can see that cartilage is not present in the bronchioles. Ciliated epithelium is present in all of them. Squamous epithelium is not present in them, and endothelium is not present in them. So correct option is B. Question number 34. The photomicrograph shows a section through a part of the human gas exchange system. Which structure is shown? Is it alveolus? No, because it contains the cartilage and the alveolus are just one cell thick. This could be bronchiole. Why? Because there we can see the cartilage are scattered. There are no C-shaped cartilage as in the trachea, so this is also wrong. And bronchus, we see there are less. Bronchus is not as thick as they have shown it here. So bronchiole does not contain the cartilage. So it is the bronchus which has the cartilage is scattered and the wall is also thick, and there are some cilia present as well. Question number 35. Which factors are required for the efficient diffusion of oxygen and carbon dioxide in the human gas exchange system? For efficient diffusion of the gases, clean and warm air entering the lungs. This has nothing to do with that. Maximized area of diffusion is affected by the surface area. Minimum distance diffusion is affected by the distance. So 2 and 3 is the correct option. Which statements correctly identify why it is difficult to eliminate TB? Humans have limited access to clean water. TB has nothing to do with the clean water. It's an airborne disease. Humans can be infected, but pathogen can be inactive. Yeah, that's correct. A number of drug-resistant strains of pathogen have evolved. This can be correct. So D is the correct option. Bacteria can become resistant to antibiotics. What can help reduce the development of antibiotic-resistant bacteria? Use specific antibiotics instead of wide spectrum. That's correct, because we cannot use any type of the antibiotic. It should be specific. Use antibiotics to treat viral infections. We never use antibiotics to treat viral infections because viruses are not affected by the antibiotics. Develop new antibiotics. That's also correct. So 1 and 3 means C is the correct option. The diagram shows one way of testing the effect of an antibiotic on bacteria. So we have the petri dish with the nutrient agar containing bacteria. These are the colonies of bacteria, and this is the disc of salt filter paper soaked in antibiotic. So the diameter of the zone measured every day for 5 days. The table shows the results of testing 5 different types of bacteria. This is the table. Zones of less than 13 mm show the presence of resistant bacteria. So which statement can be supported by this data? All the types of bacteria become resistant to the antibiotics over time. So we can see there are many where we have more than 13. So all of them are not resistant. Only type 2, 3 and 4 of the bacteria show resistance to antibiotic. Type 2, 3 and 4. We can see they have the readings less than 13, less than 13. Yeah, this option can be correct. The antibiotic can be used to treat type 1 and type 3 only, though that's wrong because they are not resistant. And types 5 of the bacteria can never become resistant to the antibiotic. We cannot say that. So option B is the correct one. Question number 39. Some responses made by cells of the immune system to a pathogen are listed. Mitosis recognizes the pathogen, produces memory cells and secretes enzymes. Which responses are correct for phagocytosis? Phagocytes do not undergo any mitosis. Phagocytes recognize a pathogen, definitely. Then only they will do the phagocytes. They don't produce the memory cells. They secrete enzymes. So 1 and 4. Sorry, 2 and 4 is the correct option. Now, last question of this paper. Some vaccines do not contain antigens. The vaccines contain a molecule of mRNA. Cells in the immune system use the mRNA molecule to make a protein antigen. The statements describe the stage of how mRNA vaccines work when they enter a cell of the immune system. B lymphocytes and T lymphocytes with complementary receptors bind to the proteins. Lymphocytes differentiate into memory cells that give long-lasting immunity. Ribosomes translate the mRNA molecule to make a protein. The cells display the protein on the cell surface membrane. What is the correct order of the stage of the how mRNA works? So first of all, we'll see ribosomes translate the mRNA molecule to make the protein. This should be the first one. Then B lymphocyte with complete receptors bind to the protein. Lymphocytes different to memory cells that give long-lasting. The cells display the protein on the cell surface membrane. Okay, this could be the second one. Then B lymphocyte with the complete receptor bind to the protein. This is the third. And then fourth one, lymphocytes differentiate into memory cells. That give long-lasting immunity. So that means 3, 4, 1, 2 is the correct option. I hope this will be helpful for you in understanding the MCQs of the questions. Thank you very much.